Assalamualaikum everyone, welcome back again to Regal Biology with Khadija. Have you ever seen a biology dissection kit and wondered what all these instruments are used for? Why are there different types of scissors, forceps, needles, and inside the single kit? In today's video, we will explore the biology dissection kit, learn the names of its tools, understand their functions, and will discuss important safety precautions. By the end of this video, you will be able to identify each instrument and explain its use confidently. Let's get started. अब डाइसेक्शन किट होती क्या है? यूँ समझ लो एक ऐसी किट जिसके अंदर कलेक्शन मौजूद है स्पेशलाइज्ड इंस्ट्रूमेंट्स का, जिसको यूज़ किया जाता है या जिन इंस्ट्रूमेंट्स को यूज़ किया जाता है प्लांट्स और एनिमल्स की डाइसेक्शन करते हुए। और यही टूल्स जो हैं ये हेल्प करते हैं स्टूडेंट्स को या रिसर्चर्स कह लो कि वो एग्ज़ामिन कर सकें किसी भी ऑर्गेनिज़म का इंटरनल स्ट्रक्चर। तो डाइसेक्शन आपको पता है बहुत ही इम्पोर्टेंट पार्ट है बायोलॉजिकल स्टडीज़ में। तो अगर हमने एनाटॉमी स्टडी करना है तो डाइसेक्शन किट को यूज़ करके हम डाइसेक्शन करते हैं और एनाटॉमी स्टडी करते हैं। तो हम चलते हैं टूल्स पे। सबसे पहला टूल जो हम स्टडी करेंगे इज़ Scalpel. It's a very small and extremely sharp instrument. Actually, this is the stand of the scalpel, and a blade should be added on the top. How can we add blade? This is the blade, the surgical blade. It comes in different numbers, 10, 11, 15, and each number has its specific roles or specific uses, and they are used to make precise and localized incisions or blade cut in surgery. How to insert the blade in the tool? Just add carefully without touching the blade, because one, it is very sharp, and second, it's sterile. And be careful while adding this surgical blade into tool. Now after adding, it will be used in surgery. And scalpel handle also vary, like number 3 or number 4. In the same way, the sterile blade, surgical blade, which is removable, is also vary in number, like 10, 11, 15, etc. So be careful. Usually in one dissection kit, two handles are present. One is for a small size blade and one is for a large size blade for different types of surgical
Section kit usually have different kind of scissors as a tool. Straight scissors, general cutting, curved scissors, and iris scissors, different kind of scissors. But here in this dissection kit there are two scissors. This one that is you are looking, one end is sharp and one end is blunt. This scissor is called sharp blunt scissor, and this one is called as forceps scissor because this is used to hold tissues or specimens during dissection. Suppose the structure is very small and you can't help with your fingers, then you will use this forceps scissor. It will help to pick up small structures that are very difficult to handle with your fingers, so you will use this to hold. Its structure looks like a scissor, but it's actually not called as scissors, it's called as forceps. In simple words, you can say artery forceps because it just look like scissor, but it locks and holds the tissues just like this. You can hold and lock the tissues, arteries. So it's not purposely used for cutting, but used in surgery and advanced dissections to handle the blood vessels, the tissues, and to grasp. So these are three kinds of scissors and forceps that are used in surgery. Other forceps are also present in dissection kit, like these that are showing in a video. You can see these are also called forceps. These are used to hold the tissues, hold the vessels when you can't do with your fingers. Well, the next is dissecting needle, and it is also called as probes. They are different in shape, some straight tips and some like blunt tips, curved or hook shaped tips, and sometimes they are lengthened point or very sharp. They have function of gently separating the delicate muscle fibers, connecting tissues and membranes, to identify, to trace, and to isolate very minute structures or organs that are underlying the organs. So this is also probe, but the shape is arrowhead and not sharp but blunt. This is also probe and used to trace or separate the delicate tissues without tearing them apart or without any damage to the tissues. And the next, this is spatula. It's a dissecting spatula. It's a flat or paddle shaped instrument. It is used to lift, spread and separate the tissues safely without damaging the delicate tissues underlying. This is also spatula, but that one is single ended, and previously was the double ended spatula. Both ends can be used as a spatula to lift the tissues or organs, and this one is single ended. This one is curved shaped teasing needle and used in mostly experiments, like while you are making a microscopic slide and you want to gently tease the muscles or tissues and separate them layer by layer, you will use this one is also a teasing uh, pipe, which is used to separate the layers or organs. And the next, this one is blowpipe or blow tube. It is simple and straight metal tube. It is used to inflate hollow organs to separate the delicate tissues by directing a gentle stream of air into them. And next is chain hooks. While you are dissecting on a dissecting tray or a wax tray, these are used to firmly anchor a large specimens onto a dissecting tray. And the next one is very commonly used dissecting razor. It's usually flat, and they are used to slice very thin uniform section of tissues while making a microscopic slide for your for your microscopic examination. When you want to make a slide of microscope, you will use this to cut a very thin layer of tissue, and then you will make a slide by using that tissue to examine. And the next is brush and a dissecting kit. It is actually used for cleaning delicate specimens, gently sweeping away any debris, so untangling blood vessels, etc. So this is the whole story of dissection kit. Whether you are performing experiment or dissection kit you are using, wear your lab coat, your gloves, handle each every scissor, sharp instrument very carefully, and never ever point sharp instruments toward yourself and your fellows. Keep the work area clean, and after ending your experiment, dispose of the biological material properly, because safety should always be your first priority in laboratory experiments. Today we have learned about scalpel, dissecting scissors, forceps, needles, probes, dissecting pins, and if you found this video helpful, don't forget to like this video, comment your answer to today's question, subscribe to Regal Biology with Khadija, and share this video with your friends.